The button, it helps. I'm Wells Messersmith, and it's a pleasure to introduce Mike Rothenberg to our Symposium series. Mike has a fairly accomplished background. He went to a small community college in Connecticut called Yale, and then went from there to Stanford, where he got his MD, PhD, and then trained at Mass General and as a resident, and then the Dana-Farber Mass General Combined Program. And he was on faculty at Mass General for a while, and then came back to Denver. He actually lives in my neighborhood. He has 3 above-average children. I can attest to that, unlike mine. And has been working at Loxo since the summer of 2015, and has quickly kind of risen the ranks and is now vice president of research and development, and is quite involved in everything from pipeline compounds, lead optimization, all the way through clinical development. So we're really excited to have him talk about developing inhibitors against uncommon oncogenic kinases. Thank you, Mike.
Can you guys hear me okay? Is it great? Thanks for the introduction. I met Wells when I was on the fellowship interview program. Wells had preceded me at MGH, and he's very famous there, as he is probably here. His name is on the primary care textbook, which I always found interesting. You might have had different interests when you started, but I appreciate the opportunity to be here. I do appreciate the compliment. I do think my kids are above average, except for my youngest, my 5-year-old Calvin, because he is an insane Patriots fan. He wears his Tom Brady t-shirt all the time. He's going to dress up as a Patriot, so he hasn't converted to the Broncos. And I see a friend of mine, Josh, from Postdoc. How are you? Nice to see you. It's been a long time. Let me get started. So these are my disclosures. I'm an employee and stockholder of Loxo, and all of our medicines are in clinical trials or development, so they're all off-label. I think everyone in this audience is involved in some way with cancer patients, whether through research, patient care, both. And this story that I'll tell you of a patient who has nothing to do with my role at Loxo, someone whose care I was peripherally involved with, for me, reminds me why I guess I get up every day and go to work. And this is a patient, a 12-year-old girl, who was diagnosed when she was 3 years old with a very advanced carcinoma in her abdomen. It wasn't clear what the histological type was. Maybe it was ovarian cancer, maybe it was mesothelioma. And for basically 9 years, her entire life, she was treated like a much older woman with advanced ovarian cancer. So she had multiple debulking surgeries. She was treated with abdominal radiation. She had multiple rounds of systemic cytotoxic chemotherapy, intravenous, intraperitoneal, with a so-called targeted therapy thrown in there without any biomarker hypothesis. Nonetheless, those treatments kept her cancer relatively at bay until she was 12. And at that time, which was the fall of 2016, this is a PET-CT scan. For the first time, her cancer had spread outside of her abdomen. Can you see the arrow yeah. So outside of the abdomen to involve her lung and chest wall. And there was really no other treatment or standard of care, treatment options available to her. And through, this little girl lives in a suburb in our state. Her mother was not in the medical community, but she was very persistent and managed really on her own to figure out how to get one of her tumor biopsies tested with a molecular testing next generation sequencing panel. Sample was sent to Foundation Medicine. I'm sure it's familiar to all of you. And the initial results came back with 2 hits, P53 splice site mutation, likely inactivating, and a TERT promoter mutation, likely leading to increased TERT expression. The problem is, and again, as all of you know, neither of these mutations is obviously targetable with an existing or experimental therapy. This was when I and several other people, including here at UC, some of my colleagues at Array Biopharma, which is a well-known Colorado medicinal chemistry country, got involved. And I was asked to take a look at this Foundation Medicine report. Buried in the back, it's about 28 pages long. On page 28 is the appendix, which includes so-called variants of unknown significance. So these are variants that have not been published before. And among those was the report of an AKT1 rearrangement. And I imagine many of your reactions is like mine, or anybody who has any sort of, does any thinking about cancer in human patients. AKT is a bona fide oncogene. There are bona fide activating mutations. Those so-called chromosomal rearrangements hadn't been described before. And there are treatments out there. There are various types of AKT kinase inhibitors. So the real question is, does this rearrangement create a activated AKT kinase? And we took a deeper look. Foundation Medicine was very generous. They provided the sequencing reads. Another Colorado-based company, ArcherDX, created a custom panel to look at whether this rearrangement was expressed at the RNA level. And all of that analysis suggests that, in fact, this rearrangement, which was expressed at the RNA level, encoded an in-frame fusion of the AKT1 kinase domain minus all of its amino terminal regulatory domains, minus its pH domain, and it was fused directly to just 13 a sequence encoding the first 13 amino acids from a gene, a protein called LAMTOR1. But those 13 amino acids were predicted to encode for a myristoylation site. And so we had a very obvious mechanism of activation, which is localization of a AKT kinase domain to the membrane in the absence of its negative regulatory domains in a location where it could phosphorylate its various substrates. And so with that finding, the search for an AKT inhibitor to treat this little girl with was on. And Genentech, there are many AKT inhibitors in the clinic, and you may be aware of some of them. There are so-called allosteric AKT inhibitors, and there are so-called catalytic AKT inhibitors. We didn't think the allosteric inhibitors would be active because the protein was missing its allosteric, the fusion was missing its allosteric regulatory sites. So we focused on reaching out to companies who had catalytic AKT inhibitors, and Genentech, to their great credit, made GDC-0068 or ipatasertib available under a single patient FDA-allowed compassionate use clinical protocol. This inhibitor is specific for all three AKT isoforms. It doesn't hit many other kinases in the kinome. It's oral, and it was in phase 2 clinical trials in adults, so we certainly knew its safety profile, its maximum tolerated dose in adults, though it hadn't been used to treat children before. Patient was flown to New York City. She was treated at Memorial Sloan Kettering Cancer Center, and she started this clinical trial with a very low dose of ipatasertib and underwent so-called sort of intrapatient dose escalation using safety and PK to guide that. These were the results 4 weeks after treatment, and I think you can see that there's been a significant improvement, a significant decrease in the metabolic activity, the FDG activity of these tumors in her lung and chest. Concomitant with that was a very dramatic, significant tumor response by traditional imaging. And this graph just shows her CA-125 levels, her tumor secreted CA-125 into the blood, and you can see that they really plummeted within the first few weeks and remained low for a while. And this effect was more than just diagnostic. I mean, she was really quite sick when she left for New York City. It wasn't clear that she would be able to go. And in a matter of weeks, she returned home to Colorado. She was present at a fundraiser that was held for her by Archer in the area. She really was like a little girl again. So a really dramatic, rapid, Lazarus-like response. Unfortunately, over the course of the intervening weeks, her CA-125 started to rise. Imaging showed that her disease was starting to progress again. A new biopsy was taken. A next-generation sequencing panel was performed to look for new mutations. None were identified. A decision was made to empirically treat her with both the AKT inhibitor and add in a rapalog, thinking about hitting the pathway a little harder. It had a little bit of a benefit in her CA-125. Ultimately, she continued to progress, and quite tragically, she ultimately succumbed to her cancer. I share the story with you because I think it illustrates all of our challenges as clinicians and drug developers. First, finding patients sometimes with a rare, oncogenic, targetable genetic mutation for which, if you had a drug or there was an existing drug, you could hope for dramatic effect on the patient's tumor and on their quality of life. Second, making really potent and specific medicines with a wide therapeutic window that inhibit only the mutation, the target of interest, ideally only the mutated target of interest and no other target in the patient's body. Finally, to think about more innovative and rapid clinical development, we have patients right now for whom we know the genetic mutations. They are dying of their cancer right now. And when you have a treatment with some expectation of safety and efficacy, can we deliver those medicines to patients like that, even if they're in early phase clinical trials or potentially even haven't gone into first-in-human trials? And of course, last, the scourge of acquired resistance. And so for the rest of my talk, I'm going to discuss 3 medicines in the Loxo oncology pipeline. 2 TRK kinase inhibitors, the first larotrectinib, or Loxo 101, which may be familiar to people in the audience here because some of its preclinical and early clinical development occurred right here in the Cancer Center. Loxo 195 is a second-generation TRK inhibitor that's been designed to overcome acquired resistance to first-generation TRK inhibitors. And if there's time, I'll finish with a discussion of a specific RET kinase inhibitor called Loxo 292 that I'll describe to you as sort of a single-step solution to all of these problems, potentially. So real quickly, I want to talk about TRK kinases. So the tropomyosin receptor kinases, or TRKs, are a family of 3 fairly typical receptor tyrosine kinases, each of which has both overlapping and unique roles in normal nervous system development and maintenance. They're involved in critical processes like pain and thermoregulation, like mood, like proprioception. Again, normally in normal cells, the signaling pathway is activated by binding of one of 4 neurotropin ligands to individual TRK receptor kinases at the membrane. Those ligands cause homodimerization of 2 receptors, intracellular autophosphorylation in the cytoplasmic domain, and the activation of the typical types of downstream signaling pathways like the MAP kinase pathway, like the PI3 kinase that you're all familiar with. In human cancer, TRK kinases are activated primarily by chromosomal rearrangements that lead to in-frame fusions of the sequence for the kinase domain of any of the TRK genes with a dimerization domain with the promoter of a gene at a disparate location in the genome. That leads to ligand-independent constitutive activity and abnormal activation of growth, proliferation, and survival. There are 2 main diagnostic challenges to bringing a TRK inhibitor, if you had one, to patients. The first is the extreme heterogeneity of the fusions with respect to different tumor types. So I've illustrated that here with both this map of an adult and a atomic map of an adult and a child. TRK fusions are found at low frequency, very low frequency, probably less than 1% in some very common cancers, like lung cancer, a few percent of thyroid cancer. They're found at moderate frequency in less common cancers, for example, soft tissue sarcomas or melanoma. And they're quite common. They're actually the defining pathognomonic genetic alteration in extremely rare childhood cancers. So examples are infantile fibrosarcoma, congenital nephroma. But you can imagine that to find patients, theoretically, you'd have to have a screening methodology and screen patients with many different types of cancers, adults and children, to find what are essentially potentially low-frequency alterations. The second feature that makes bringing a TRK inhibitor to patients challenging is the complex genetic architecture of the fusion. So there's 3 TRAC genes, again, TRAC A, B, C, or NTRK1, NTRK2, NTRK3. There are multiple, maybe 100 different fusion partners that have been identified. The introns from the TRAC genes that mark the fusions are quite large. So many existing sort of push-button next-generation sequencing modalities don't actually cover all of the sequences. They can't identify all of the TRAC fusions that have been identified in patients. One approach that we and others are taking to this problem is to develop an IHC assay. So this is very similar to the approach with ALK fusion cancers, where I think, as you know, an IHC assay has essentially replaced FISH or other NGS assays to identify ALK fusions. It requires that normal TRAC kinases are not expressed at detectable or high levels in normal tissues. But we are currently pursuing this as a way at least to initially identify these patients. The test may be sufficient on its own. We're combining with NGS to see if this would be a way to get through the diagnostic challenge of finding these fusions. So that brings me to larotrectinib, which is also called Loxo 101. We believe it's the first and only selective TRAC inhibitor in clinical development. It's highly potent for each of the TRAC kinases, TRAC A's, B, and C, and their fusions, with essentially low single-digit nanomolar activity in cell-based assays. It's very selective. It's very orally bioavailable. It has really good drug-like properties. And its clinical development has been relatively fast. So the first patient was treated right here at UC Denver in March of 2015, the first patient with an actual TRAC fusion. In July of 2016, so a little more than a year later, the medicine received breakthrough therapy designation from the FDA, and the pivotal enrollment, the enrollment that we hope to look at and analyze for efficacy in TRAC fusion cancers, was completed in February of this year. So I'm sure you're all familiar with these kinome renders or kinome dendrograms. And what I've done is to show the dendrogram of targets for a variety of kinase inhibitors with some degree of anti-TRAC activity. And again, just to be sure we're all on the same page, about 550 kinases in the human kinome. They're shown here by their sequence and evolutionary relatedness, and the end of each of these little branches is a different kinase. And you can see that all of these inhibitors inhibit a lot of kinases, so I've colored those with the red circles or the green circles, in addition to the TRAC kinase, which I've shown in blue. I've left out TRAC-C because we've actually done these experiments, and it's a commercially available screen, and TRAC-C isn't in the assay. But you can see that there's lots of circles in addition to the blue ones. And compare that to Loxo 101. It's really 1,000-fold more potent against the TRAC kinases, each of them, than it is than any other kinase or even non-kinase target. And that's both in sort of enzyme and cell-based assays. So it certainly has a very different profile with respect to selectivity. This slide summarizes the overall larotrectinib clinical development program. There are actually 4 ongoing clinical studies that make use of the medicine. The first study was an adult phase 1 study, which was a traditional phase 1 study in patients with any advanced solid tumor. One didn't need to have a TRAC fusion to be enrolled. There is an adult and adolescent phase 2 basket trial ongoing that enrolls patients with identified TRAC fusions identified locally to one of 8 tumor type baskets. There's a pediatric phase 1/2 ongoing, again, for patients with solid tumors. They'll really focus on patients with TRAC fusions. And actually, larotrectinib is the TRAC inhibitor that's been selected for NCI match. We think, is sort of the large NCI-based basket study in which patients are entered onto cohorts based on tumor genotype to particular therapies that are thought to be active against that genotype. And again, the overall development strategy was not that unusual, with the typical phase 1, phase 2, and with typical primary and secondary endpoints. So the initial clinical activity in patients with TRAC fusion cancers across those 3 studies was recently presented at the ASCO annual meeting. And so among 50 patients who had had confirmatory radiologic imaging, The overall response rate was 76%. Again, these are only patients with tract fusions, including a 12% complete response rate, slightly increased when you included 5 more patients who had had their initial scan at the time of the data cutoff but had not undergone their confirmatory scan. So really a very active medicine, the kind of activity you hope for and that you've seen with very active drugs in very oncogene-addicted cancers. So think of EGFR mutant lung cancer and EGFR inhibitors, or think of ALK inhibitors and ALK fusion lung cancer, et cetera. This is the waterfall plot, which is sort of showing you the best response among the 50 definitive patients who had confirmatory scans. And you can see that the majority of patients had confirmed partial responses, including some complete responses. And the thing that makes larotrectinib, or really what makes tract fusion cancers different from the examples of EGFR lung cancer or ALK lung cancer, is really the following. So that activity is completely independent of tumor type. So these 50 patients were spread among 17 different tissues of origin, tumor types, from lung cancer to childhood sarcoma, to GI stromal tumor, to pancreatic cancer, to melanoma, and so on. And the activity was really independent of that tissue of origin. It's independent of age, so with responses in and pretty long duration of responses in children, infants, in adolescents, in adults. Independent of the tract gene, independent of the fusion partner. And again, median PFS has not been reached, and the duration of response, again, is the type of response hasn't been reached, is the type of response that you hope for a medication like this. And just to give you sort of 2 examples, so this is Bob Doble's patient, so the first patient with a tract fusion cancer treated with larotrectinib. When she came to our attention and Bob's attention, she was 42 years old. She had a very advanced chemorefractory undifferentiated soft tissue sarcoma that had progressed through multiple cytotoxic chemotherapies. She was very symptomatic, on oxygen, with dyspnea. This is her CT scan of her lungs before treatment. I think you could fairly describe as innumerable cannonball-like lung metastases. And these are the scans after 2 months, 8 weeks of 100 milligrams twice per day dosing, 1 pill twice per day. I don't think Bob is here. He's not here, right? But Bob will tell you that clinically she improved within days, within the first week of starting therapy with, near complete resolution of dyspnea, resolution of hypoxemia off oxygen, and scans really correlated the depth of that response. She's actually still on treatment. I mean, she's been on larotrectinib for more than 2 years. Again, she was treated towards the middle of 2015. So, really an amazing, remarkable, persistent response. And just to show you the other end of the spectrum, and not meant to be gross or anything, but this is a little girl, 14-year-old, who may be the world's first patient with metastatic secretory breast cancer. Secretory breast cancer, or juvenile secretory, I should rephrase that, juvenile secretory breast cancer is a very rare breast cancer histology that portends a good prognosis. There may have only been 12 or 13 cases reportedly in the world. They're actually known to have NTRK3, so TRK-C gene fusions, ETV6 NTRK3. This patient was born and lived about 400 miles outside of Dhaka in Bangladesh and came to our attention almost miraculously through a internet-based global tumor board for which a member of our SAB was on. We obtained tissue to determine whether it had a tract fusion. It did. She was brought to New York City to be treated, and I think her response speaks for herself. She had just a fungated lesion that had broken through her chest wall, and with days it had essentially disappeared. And again, she was all treated on one of these more novel, single-patient, FDA-allowed, sometimes called but not really compassionate use protocols, so that we could get her treated at an efficacious dose as quickly as possible. So I'm going to shift gears and dive right into the scourge of acquired resistance. So I think, as all of you know, acquired resistance to targeted kinase inhibitors is essentially universal. It accounts for the disappointingly short progression-free survival for patients despite the dramatic initial responses. Biochemically, or from a signaling perspective, the types of mechanisms are grouped into in the kinase, so second mutations in the targeted kinase itself that increase its activity, that directly interfere with drug binding, and so-called bypass tracks, so activation of another receptor tyrosine kinase, making the cell dependent on both for survival and thus requiring inhibition of both to have an anti-tumor effect. The first generation TRK inhibitors were identified in 2016. They were actually identified in patients who had responded to and progressed to a different kinase inhibitor called entrectinib. Entrectinib, or RxDX101, is a multi-kinase inhibitor with activity against a variety of kinases, including potent activity against the TRK kinases. And both of those mutations were in the kinase, in two regions of the protein of the kinase domain called the solvent front and XDFG motif. And as you know, it can take years to develop next generation kinase inhibitors that overcome resistance. So think about the example of CML and BCR-ABL and the T315I gatekeeper mutation, or the EGFR T790M gatekeeper mutation. I mean, it really took 10 years to go from imatinib to ponatinib, and even ponatinib isn't a great solution because of its toxicity, or to go from erlotinib to osimertinib. You know, ALK, a little bit shorter, 5 years to develop alectinib, or ceritinib after or brigatinib after crizotinib. But the real question that faced us, because we knew patients were progressing, was, can that time interval be shortened? Can we identify and deliver a new medicine to the patients who are actually treated on our early clinical trials at the moment that they progress, were they to progress? At the time, no one had actually progressed on larotrectinib. Not that we didn't expect that to happen. And so, together with our collaborators at Array BioPharma, and I should emphasize that really all of our work is done through a very close and productive collaboration with Array, again, based here in Colorado. The approach that we took was to essentially solve the crystal structure of the TRK kinases with the resistance mutations and look in silico at the potential effect of those mutations on drug binding. And so here you see the crystal structure of either TRK or TRKC with the so-called solvent front mutation in blue, or the XDFG mutation in red. And it was obvious how these mutations engendered resistance. They directly interfere sterically with the binding of here larotrectinib. I don't know how well it's seen, but I've tried to circle the steric interactions, which are shown with dotted red lines. One of the advantages of having worked so long and so hard on TRK kinases in general is that we actually had a very rich and chemically diverse lead compounds from which we had selected larotrectinib from several. We had 4 chemical series, completely structurally different. And so really for us, it could be a matter of, minimal chemistry is sort of just doing screening. We did an in silico screen, and we identified a potential hit called Loxo 195, which you can see, at least based on our structural modeling, looked like it could bind to the A to B binding site. It was small enough, it was compact enough to avoid the potential steric interactions of these resistance mutations. So Loxo 195 is the second selective pan-TRK inhibitor in clinical development. It has this unique structure. It's actually a macrocycle. It's a closed circle, which makes it very compact. It's highly potent against wild-type TRK, TRKB, and TRKC without or with any of the acquired resistance mutations that have been identified in patients, as well as other mutations that had been identified preclinically. And we believed it would have larotrectinib-like selectivity and really promising drug-like properties. And so to show you the developmental timeline, again, first patient with acquired resistance to this other TRK inhibitor, entrectinib, were identified in 2 patients in November of 2015. And in October of 2016, less than a year later, we began treating the first true TRK fusion cancer patients who had developed acquired resistance to larotrectinib. So less than a year from an idea to a clinical to really a clinical trial. And again, both patients had been treated, had responded to larotrectinib, and then had progressed. Their tumor had been sequenced, and we identified these resistance mutations. So this work was recently published in Cancer Discovery, and I refer you to that manuscript, and I'd be happy to talk to you about it later afterward. This is just sort of a summary of the types of preclinical that we did once we had identified a lead. These are cellular IC50 curves, actually looking at phospho-TRAC levels in cell lines engineered to express either a activated, if you will, wild-type TRAC-A or an ETV6 TRAC-C fusion without or with the two classes of acquired resistance mutations. I think you can see in all cases the red curves, which are Loxo 195, are shifted to the left, so they maintain essentially near single-digit nanomolar activity, low nanomolar activity despite the presence of these mutations, while larotrectinib really loses significant activity. These are the in vivo efficacy curves using mouse allografts. As expected, both larotrectinib and Loxo 195 cause tumor growth inhibition in either a human cancer cell line expressing, again, a TRAC fusion without an acquired resistance mutation, or an engineered 333 cell line expressing an activated TRAC-A. But in engineered lines in which the activated TRAC-A possesses an acquired resistance mutation, you can see that only Loxo 195 maintains significant anti-tumor activity. And as a slightly different way to think about selectivity, we screened a large panel of human cancer cell lines of a variety of tumor genotypes with Loxo 195, just using cell counting, cytotoxicity as a readout. And what you can see is that the cellular IC50 for we did this in a blinded fashion, but the cellular IC50 for 3 human cancer cell lines, each of which is addicted to an endogenous TRAC fusion, is really in the single-digit nanomolar range, and there's virtually no inhibitory activity, out to 10 micromolar against 84 cell lines with a variety of tumor genotypes and a variety of tissue types, but unified by their lack of a driving oncogenic TRAC alteration. So I'm going to shift gears and talk to you about the early clinical development of Loxo 195. And I wanted to start with a reminder of the traditional way that first-in-human clinical trials are performed in cancer patients. And as you know, this traditional way was really developed in the era of cytotoxic chemotherapy, where we often didn't have biomarkers. And as you know, in general, there are a variety of designs, and I think that's less important than the overall themes. But the general approach is to start with a population of advanced cancer patients who are not pre-selected based on a biomarker, but may contain a few patients in blue who ultimately have a biomarker, a genetic lesion that will lead them to respond to the therapy, and to treat them, 3 patients at a time, to treat them at a starting dose that is determined by the signal that is seen in preclinical toxicology studies and then divided by 10 according to FDA regulations. Show that all 3, or at least no more than 1 of 6 patients, tolerate the medicine before you enroll a new 3 patients at the next dose, and go stepwise till you get to a maximum tolerated dose, till you get to a dose that you think is active. And at that point, a decision may be made to focus in on patients most likely to benefit. And so you do what you might do a separate phase 2 study, a phase 1b, a dose expansion. And this approach clearly works. It's very effective in the era of cytotoxic chemotherapy. It's continued to be used for targeted therapies, but it certainly risks treating a lot of patients up front who are not destined to benefit and missing an opportunity to help patients who would have responded were they treated at a high enough dose to cause a high enough exposure to have anti-tumor activity against the target. A different approach, and one that we've used and others have used, is called a variety of things, but one term to call it is PK-guided accelerated phase 1. And in this approach, we only treat patients who we think are destined to respond, who have the biomarker of choice, who have a TRAC fusion with an acquired resistance mutation. And those patients are treated at a starting dose, and we obtain blood samples during treatment. And we do what's called semi-real-time PK analysis. We get PK samples after the first dose, maybe after a few days on dose. We measure the PK. And if the patients tolerate treatment for a week or 2 weeks or however long you want to do it, but the exposure is lower than you think you need, those patients' dose is escalated, so-called intrapatient dose escalation. And we repeat the process iteratively. This way, you're getting every patient to a potentially efficacious dose, again, guided both by PK, but still guided by safety. And you're only treating patients who have the potential to respond. And the way, so we utilize this approach for the first 2 patients treated with Loxo 195. And again, this is another way to look at it, but essentially we identified a starting dose based on preclinical experiments, cut that dose, by a significant amount, start the patient at that dose, do real-time PK on days 1 and 8, measure safety, and if the medicine is safe and the PK is below what we're aiming for based on some pretty good, albeit preclinical science, those patients undergo a dose escalation and you repeat the process. You repeat the process till you're in the exposure target range. Obviously, if you repeat the process and lower the dose, if there are toxicities. Otherwise, assessment of safety and efficacy is the same. And so again, and this is work that was already published in this manuscript. So here are the first 2 patients that we treated. The first patient was an adult patient with an NTRK1 rearranged colorectal cancer that had progressed despite multiple prior chemotherapies, had none of the other sort of canonical genetic mutations that you come to expect for colorectal cancer, so likely to be a TRK-dependent cancer. This patient had been treated on our phase 2 with larotrectinib and had a confirmed partial response that lasted for 6 months. She then progressed, and analysis of a biopsy of a recurrent progressive tumor showed a TRK-A G595R solvent-front resistance mutation. The second mutation was a little 2-year-old girl who had an ETV6 NTRK3 rearranged infantile fibrosarcoma of her jaw. She had been treated with multiple prior surgeries and chemotherapies. She was treated on the phase 1 study of larotrectinib. She had 90% tumor regression that lasted for 6 months. She then recurred locally, and a biopsy showed a homologous solvent-front G623R mutation in the TRK-Z kinase domain, so the homologous position in the solvent front. This is a complex slide, so I'm going to take a couple minutes and go through it. So what I'm showing you is the actual real-time PK data for each of these patients on larotrectinib and Loxo 195 because we measured it during their treatment. So let me focus in on this leftmost graph to explain it, and then I'll quickly go through the remaining graphs. So this is the profile of this adult patient treated with larotrectinib when the only thing present was the fusion. There was no acquired resistance mutation. So each column is a different TRK kinase genotype. So her genotype when she started treatment, TRK-A, her acquired resistance mutation, for what it's worth, this other XDFG mutation. And the y-axis is drug concentration. And so what you can see is that at a dose of 100 milligrams twice per day, this is actually her minimum concentration at steady state and her maximum concentration in steady state, this vertical line with the 2 circles. And again, this is just absolute drug concentration in plasma. And what I've done here is each rectangle is showing you on that scale the drug levels of larotrectinib required to achieve either TRK-A kinase IC50 inhibition, that's literally just based on in vitro cell-based phospho-TRK inhibition, just on our preclinical data, or so-called IC90, 90% inhibition of TRK-A. We've simply taken those IC50 and nanomolar and converted them to plasma levels of drug in a patient. And so what you can see is that the actual drug levels in this patient initially were sufficient to cause pretty robust inhibition. I mean, they're getting well above the LMNA and TRAC1, TRAC-A IC90 at her dose, really for the duration of the dosing interval. But they're really insufficient to cause significant inhibition if that TRAC-A kinase harbors either a G595R solvent front mutation, which he developed at acquired resistance, or if it happened to develop this other resistance mutation. So is that clear? Great. So now look at the same profile now with real-time PK on Loxo 195. And again, here we're projecting the IC50 and IC90 of each target based on the in vitro anti-cellular activity of Loxo 195 rather than Loxo 101. So this was her starting dose, you see here, where she was getting reasonable coverage, but certainly her steady state Cmin or trough levels were sort of below what you might want and what we wanted. We really were looking for sort of close to IC50 at trough. That was our PK trigger, if you will. So she underwent intrapatient dose escalation, 50 to 100 milligrams twice per day, and at least based on PK, now you're talking. We're now at a level where we're hitting her particular acquired resistance mutation very hard for most of the dosing interval. I'll just quickly say you essentially see the same pattern here. On larotrectinib, this patient had good PK pharmacodynamic activity against her endogenous ETVC and TRAC3 fusion, but really not against the acquired resistance mutation that she developed. We did intrapatient PK dose escalation till we got to a dose that looked to us like it was going to cause pretty potent inhibition of the acquired resistance mutation that she developed. So, as it turns out, there have actually been six, at the time that we released this data, there were 6 patients identified who responded to larotrectinib and then progressed. And interestingly, 5 of 6 of them all had the solvent front mutation that we were confident Loxo 195 would be active against. It's what the PK suggested, and it's actually what the clinical benefit suggested. So this is a fused CT, a PET image, a metabolic image of a liver metastasis, and an intraperitoneal metastasis in this patient after acquired resistance to larotrectinib, before she was treated with Loxo 195, and this is 2 months on therapy when you can see, essentially complete resolution of that metabolic activity, that FGG, and dramatic tumor response. And she's actually still on medicine, so she's now benefited for a longer time from Loxo 195, the second generation inhibitor, than she did to Loxo 101. And then this is an image of this, really disfiguring, terrible jaw sarcoma in this little 2-year-old girl. It's really anatomically hard to define, but I think you can see the anatomic abnormality here again before treatment and then after 2 months of Loxo 101. And she's really had just significant dramatic response. You can start to appreciate, the carotid and her neck vasculature. Now, she actually ended up benefiting from Loxo 195 about as long as she did from Loxo 101. Sadly, she was doing very well, and she did have a confirmed partial response, but she ultimately was hospitalized with respiratory distress. She was really quite sick. She had pleural pericardial effusions. We're trying to understand what happened, but ultimately she did succumb to her cancer. The silver lining that she got about as much benefit from the second medicine again as the first medicine. So I think I'm supposed to finish at 12:50, so I have 5 minutes. So I think I can finish this up. So now I'd like to shift gears, and I hope this is not getting boring or repetitive, but I'd like to talk to you about the medicine that is most dear to my heart and one that I think is cause for excitement and has some similarities and differences to the TRAC inhibitors, and it's called Loxo 292. It's a RET inhibitor. So just to remind you, RET is activated in human cancers primarily by 2 genetic alterations, either activating mutations, extracellular or in the kinase domain, that essentially have the effect of directly activating the kinase. Those types of mutations are predominantly found in a relatively uncommon subtype of thyroid cancer called medullary thyroid cancer. And then there are chromosomal rearrangements like TRAC, like ALK, like ROS, causing oncogenic hybrid fusion proteins that have been identified in a reasonable frequency of patients with papillary thyroid cancer. I think most relevant for potential benefit, about 2% of patients with lung cancer, 2% of patients with lung adenocarcinomas, and then sporadically and relatively rarely in other tumor types like colorectal cancer and breast cancer and some chronic myeloproliferative diseases or MDS. You may ask why develop a RET-specific kinase inhibitor, and the reason to ask that question is there are actually several multi-kinase inhibitors in the clinic right now with at least some degree of anti-RET activity preclinically. And I think these are medicines that you're familiar with, so cabozantinib, vandetanib, lenvatinib, alectinib, ponatinib, so the list goes on. Some of these are approved for other cancer types. They're not approved for patients with RET alterations. Cabozantinib and vandetanib are approved for medullary thyroid cancer, though not in any type of genotype-directed way. There's some evidence that they're more active against the RET mutant than the RET wild-type MTCs. Our thesis is that the efficacy of each of these inhibitors is limited by insufficient RET inhibitory activity in patients, by toxicity from stronger inhibitions of off-targets. The most well-known to you is probably KDR, VEGFR2, the VEGFR2 tyrosine kinase, and poor PK. And I think the clinical data supports that it is a target. There are moderate responses, limited progression-free survival in patients with RET-dependent cancers, but there's significant toxicity. And our thesis was that we could do better with a more selective inhibitor. So that led us to Loxo 292, which was really developed like the TRAC inhibitors. And it has the features that preclinically it's very active against the really all of the diverse types of RET alterations in cancer. So diverse RET fusions independent of the fusion partner preclinically, KIF5B RET, CCDC RET, NCOA4 RET. We've looked at several of the common ones. And it's active against the diverse types of activating mutations found in medullary cancer. And again, an approach that we did differently is we tried to think about potential acquired resistance mutations to existing kinase inhibitors, and we developed it to be immune to those, and the best example of those would be a gatekeeper mutation. That's substitutions at V804 in RET. These are actually found at low frequency in hereditary medullary thyroid cancer. It's the gatekeeper. It's like EGFR T790M or BCR-ABL T315I. Hasn't been shown yet to occur in patients treated with multi-kinase inhibitors who have benefited and then progressed. But we figured it was reasonable to try to have a medicine that would be immune to that should they occur. It's very selective, and it's got very favorable pharmacological properties. So, again, this is kinome selectivity. I've listed a bunch of multi-kinase inhibitors in the clinic, either that have potential anti-RET activity or are being used in these patient populations. And I think you can see that they all hit a lot of targets other than RET, which I've colored in green. Loxo 292 has a larotrectinib or Loxo 195 profile. It's really very selective for RET and its mutations and fusions. Again, just some preclinical experiment that I'm just going to name but skip over the details. So it's active against in vivo in animal models of tumors driven by KIF5B RET. It's actually active against tumors driven by CCDC6 RET, even a RET fusion that contains in the fusion a V804M gatekeeper mutation. So the blue are the efficacy curves for Loxo 292. Red and green are multi-kinase inhibitors, cabozantinib and ponatinib. And preclinically, it's active in the brain. And so these are survival curves for animals in which a RET fusion PDX has been injected into the brain. We wait some time and then start treating with water, and all the animals succumb to their disease. We use ponatinib as a control because ponatinib is advertised to be highly brain penetrant. It works a little bit, but not well. Loxo 292 looks like it kept almost all of the animals alive for the duration of the experiment. All right, so I'm going to speed up and just tell you that we've initiated a first-in-human phase 1 clinical trial. It's a typical phase 1 trial. It's now open at 10 sites globally. The first patient was treated in May, and recruitment is ongoing. We're in dose escalation, It's a fairly traditional design with the caveat that we treat all advanced solid tumors first. You don't need a RET alteration, but once we have exposure that we think suggests to us we should be hitting RET hard, we narrow to only patients with RET alterations. And I'm going to just briefly finish that actually we'll be presenting the first early preliminary clinical activity for this medicine at the World Congress on Lung Cancer annual meeting in Japan next week. We'll describe the first 2 patients with RET fusion lung cancer treated with Loxo 292. One has a CCDC6 RET fusion lung cancer who actually responded to the experimental multikinase inhibitor RxDX105, but then progressed. And the second is a woman with a KIF5B RET fusion lung cancer who developed progressive brain metastasis on a brain-penetrant multikinase inhibitor that is thought to have some anti-RET activity. And the data that we'll present is that the first patient had a confirmed partial response on the very first dose level of Loxo 292, the lowest dose level, 20 milligrams once per daily, and she remains on treatment. And then the second patient, so this is just a cross-sectional image of her chest before starting the medicine. And this patient was actually treated on one of those single-patient protocols with intrapatient dose escalation because she was symptomatic from her brain metastasis, so she wasn't eligible for the clinical trial. So she started on a dose of 20 milligrams twice per daily and has been escalated much higher than that, has tolerated it very well. So I think you can see her lung disease before treatment. This is her lung disease after treatment. She's had a significant unconfirmed partial response. We'll update those data at the meeting next week after 2 months of medicine. She remains on therapy. And this is her brain before Loxo 292, and I think you can see all of these metastases, and this is her brain on Loxo 292, and she's really had a very dramatic intracranial response as well. Her symptoms actually improved within the first week of treatment. She had short-term memory loss, and she had trouble walking. She had a gait disturbance, and this improved significantly, and she's back to normal. So I've overused my time, so I'll just finish by reminding you of our challenges, and I hope you've seen some of the ways that we, and I imagine all of you, are working to overcome the challenges of patients who may have genetic lesions that could lead to a rational targeted approach that could offer benefit. And just finish with my acknowledgments, really primarily to the patients, families, and caregivers who have been treated with Loxo Oncology agents and our very close collaborators, Array Biopharma. And I'm sorry for going over a little bit, but I'll take any questions if you have.
What is the effect of Loxo 195, the second generation, on the normal mutations before the second?
Yeah, I mean, preclinically, it's equipotent. It may be a little more potent. So it's not mutation specific. It inhibits the TRK kinases as well.
With that, is there any concern that you're then sucking for an even more aggressive mutation by you're attacking the first mutation and the second line mutation at the same time? So now you're forcing a third line mutation out rather than simply saying go back to the first mutation.
Yeah. So the question is, is this sequential? I think another way to ask your question is sequential treatment worse than maybe using Loxo 195 up front, or will acquired resistance develop to Loxo 195 as well? So,
I don't know if you're familiar with the work of Robert Gattenby, where you'll use an inhibitor, and the second when a new mutation comes out, you go to the inhibitor for that, but it has no activity against the original mutation. So then essentially you're forcing the cancer back to the baseline mutation.
Sure. Okay. So I understand. So I think your larger question is, can you get creative and effective ways to dose these medicines? So you're really limiting your medicine to the, to the specific clone that is dominant in causing the patient's symptoms, and sort of saving your best agent, kind of keeping it in your pocket. So I think that's an excellent question, and I mean, we could talk for hours about that. I'll simply say that I think that we do need to think about innovative ways in clinical trials to do those types of approaches, and with the ability to detect circulating free plasma DNA, we may have that. One is somewhat constrained by what's acceptable, ethically and from a regulatory point of view. I can't say much about the activity of Loxo 195 at this point, but we don't have evidence that those tumors are more aggressive. We do think that they're still addicted to TRK signaling, but could they develop another mutation? Of course. But if we're starting to string together therapies, think of erlotinib, osimertinib, think of alectinib, think of crizotinib, alectinib, lorlatinib, maybe going back to crizotinib. I think we are ultimately, by stringing together the best therapy we have at the time that we're treating the patient, we may have the greatest impact. But your point is an excellent one, and we have to think about how to do that in a patient. So, another question?
So I'm a colleague of Bob Neville's, and so he's been telling me that KIT5B threats are rather insensitive to the TKI resist state. So your data looks like your drug's equally effective on KIT5B.
So all I can say is, I think the image speaks for itself. This is a woman with a KIT5B RET fusion with resistance to alectinib who is extremely sick, and she's not only clinically improved, but she's essentially had like a dramatic response in the lungs and in the brain. So, look, I'd love to discuss. So the question, and I'm sorry, we've met before, but it's Lynn, that's right. So, the question is there's a question, there are different fusion partners for RET, and actually many different kinases, and there's some preclinical data and maybe some clinical data that some fusion partners are less dependent on the fused RET kinase or otherwise less sensitive to the fusion. And I've heard different explanations for that. Remember, these fusion partners are bringing along their promoter. Are they more highly expressed? I don't buy that explanation. If you think about, we're talking about an enzyme and we're talking about a small molecule whose concentration is far in excess of any protein. Is it localized differently? I think there's interesting speculation. All I can tell you is that, and I should remind you, and you're right, this is an N of 1, but we don't see it preclinically in our experiments. We looked at KIT5B RET, and we definitely don't see it in this patient. There's no question that this patient had a pretty dramatic response, so hard to say. It's a good point. I'm not totally convinced by the model, but I don't know all of the things that Bob is doing, and I'm sure he's been very thoughtful about it. In this patient, we don't see that. So, question?
As you mentioned, one of the first fiber resistance system is to develop the gatekeeper mutation. In your NTRK patients, you have the solvent clone mainly or only. Is Loxo 195 can they target the gatekeeper mutation, or that will be another resistance that your patients can develop?
Yeah, so that's a great question. We asked that question. Actually, we worked with Bob early on to sort of preclinically evolve resistance to Loxo 101. And Bob has a nice poster from, I think, the AACR Molecular Targets meeting from 2015. And if I'm remembering correctly, there are actually two other mutations, one of which was a gatekeeper. And so we tested our medicines against that mutation, and Loxo 195 is very active against it. It hasn't been published to have, to my knowledge, it hasn't yet been appreciated in patients. So were a gatekeeper to arise, we do think that 101, as well as the other kinase inhibitor, larotrectinib, which I slip into 101, would be susceptible to it, but not Loxo 195. All right, thank you. Oh, good.